I'd like to welcome everybody to the February 16th meeting of the iMac MSM Working Group on Multiscale Modeling and Viral Pandemics. We're very grateful to our speaker for agreeing to talk to us today. As always, I need to remind you that these meetings are live streamed, recorded, and publicly available for future viewing. Tom Helikar and myself are the current co-leads of the group. Jib Sluka, Lorenzo, and Joel are senior administrators helping us out with everything. Please contact us if you have any questions, suggestions, comments, criticism. We have our usual communication channels. They would work better if we use them more. The wiki page does seem to be being updated. Please remember to send us your successes, press releases, published journal articles, opinion pieces, anything that you would like to have highlighted on the wiki page, job advertisements also. We have our growing list of YouTube videos. Please help us make those available as much as possible. And we do have a LinkedIn page if you're not a member. I think we have about 40 people following the LinkedIn page. We could definitely use help getting that out there. There's also a Facebook page, which hasn't done so much. Jib created this handle MSM Viral as an annotation. People can try using that as a tag for related announcements. There are quite a few announcements to make today. First is that Reinhardt and Anna's big workshop in Saclay will be the 15th of May till the 2nd of June. Applications are now open for that. Please contact Anna and Reinhardt for more information. And if you need any help getting into it, I know I had trouble registering, so I needed some additional hand-holding on that. So Reinhardt, I'll turn it over to you for other comments.
Well, I would just second your comment that unfortunately there have been a number of issues in registration. So, for example, if you submit incomplete information for some reason, then your application gets automatically rejected without necessarily telling you why. So don't be discouraged. If that happens to you, please email Anna or me, and we'll get it fixed. It should be an exciting workshop. Lots of people have already agreed to spend time there. So if you're interested and if you have any questions, talk again to me or Anna, and hopefully we'll see you in Paris.
Thank you. It's worth the trouble to register, I promise. Okay. The second thing is the iMac MSM meeting is finally going to happen after many years of discussion. It will be June 28th and 29th in Bethesda. And there are going to be major initiatives in that meeting discussing model sharing, digital twins, standards, and the way forward. And there are a number of organizers, but Herbert Sauro is one of them. And so if you have ideas and comments for that, please contact Herbert for more information. So we hope that people will be there too. It's going to be a busy summer of in-person meetings, I'll say, quite a few. I need to say on my own behalf that Indiana University, Bloomington, is announcing 2 open rank, those could be full professor positions, in the technology or research related to next generation biomedical digital twins. They'll be housed in our Department of Intelligent Systems Engineering. There is a job posting available. If you want to nominate yourself, your friends, or ask me more about it, I'm always happy to talk to you about it. So that is an initiative out of Bloomington. There also is an initiative in UBC. Their Resilience Institute has a very large initiative, 2 hires at the junior level, 2 hires at the associate professor level, and 2 hires at the senior level in the topic of biological resilience. And while that I think is primarily focused there at the experimental and clinical directions. They seem to also have some significant interest in bioinformatics, machine learning, and computational modeling. And so I encourage people to take a look at that opportunity as well. It looks like really quite a remarkable effort that would be very relevant to the people here in this working group. We have our steering committee meeting as usual tomorrow morning at 11 a.m. We're going to be discussing the Blueprint initiative, and we would love to have your opinions on that. I will be leading that tomorrow because Thomas will be traveling. And I guess I should open the floor now to any other announcements from the community. Actually, there are more announcements today than usual. There's quite a bit going on, I think.
Yeah, maybe I can mention a workshop, I guess one could call it, as part of the summer meeting of the Society for Math Biology. This is organized together with the NIH, the NIAID Institute, on essentially what one could call the state-of-the-art within host modeling. The effort will be to bring largely clinicians and immunologists to talk about things rather than modelers. There'll be some modelers as well, but it'll be an interesting event that takes place one afternoon and one evening.
Okay, thank you, Reinhardt. Any other announcements? So, as I say, a lot going on. It's great. Okay, coming up in 2 weeks. Question: is the Bethesda meeting by invitation only? I think that the Bethesda meeting is reasonably open, but you should probably contact, if you're NIH funded currently, which most people I think in the room would be, I think you would qualify. But I think it's generally pretty open. I don't know what the registration rules are. Grace Peng would be the person to talk to about that. In the past, those iMac MSM meetings have had a lot of people who were not only iMac MSM members. Going forward, we have Maria Abu-Chakra from University of Toronto, who's in charge of a very large Canadian digital twin effort in the context of embryological development. And then we have a series of speakers lined up: Michael Benson from the Karolinska on the Swedish Digital Twins Initiative, the rescheduled meeting of Julio Sanz Rodriguez. We still have an open slot, I believe, on March 30th. So if you would like to speak, you have students or postdocs would like to speak, or if you have suggestions for speakers, please let us know. You could either fill out the Google form, and maybe Jim's, you could paste that tiny URL in the chat for people while we're talking, and also maybe Lorenzo's email address if you'd like to suggest speakers. Again, this is, he's been, oh, sorry, yes, there's a typo in here. Thank you very much. So we don't have a slot on the 30th, it's twice. My mistake. I read it as April 30th. Thank you. So actually we have four meetings scheduled going forward. There always are opportunities, so please let us know who you'd like to hear. The suggestion link is in the chat if you would like. Okay, I don't want to take any more time from our speaker. We're very pleased to have Pras Pathmanathan from the US FDA, who's going to be talking about credibility assessment of patient-specific computational models.
Okay, hello everyone. Can you hear me okay? Okay, good. I've been told that sometimes recently my I've been having issues with my mic and it's been cutting off or making me seem like I've gone far away. So if that happens, please let me know. Okay, let me share my screen. Assume you can see my screen okay? Okay, great. All right, yeah, thanks again for inviting me to do a talk here. This is a talk on a recent paper that we published last year, focused on credibility assessment of patient-specific computational models. And in the paper, we approached this topic by looking at cardiac models, looking at cardiac modeling as an exemplar field. So it's not, I'll say from the beginning, nothing to do with immunology, but patient-specific modeling. And hopefully there'll be some generalizable insight into patient-specific models that will apply to digital twins and patient-specific models that are developed for other applications, such as immunological applications. So start off with a quick disclaimer. I should have maybe introduced myself, by the way. I'm a scientist in FDA in the Center for Devices and Radiological Health. That's the silent D in FDA devices, and I'm in the research office there, Office of Science and Engineering Labs, and part of our regulatory science program on credibility of computational models. So to a quick disclaimer, mention of any commercial products in this presentation should not be construed as an endorsement by HHS, and these are my personal views and opinions, not those of FDA. So I'll start by talking about how modeling and simulation is relevant to medical devices, and this slide tries to categorize different types of ways that modeling and simulation is relevant to medical devices so that we can start to understand where I'm coming from, where we're coming from in terms of credibility assessment, and where patient-specific models fit in. So 4 different applications here of different types of ways that simulation could be used in a regulatory submission. And on the left-hand side, there's the most established approach, which is in silico device testing, where a computational model of a device is developed and used to generate safety or effectiveness data that's either supplements or replaces traditional test data such as bench, animal, or clinical. So here there is a real device out there in the real world. We model it, and it could either be a model just of the device, or it could be a like this that could replicate a bench test, or a model of a device that's embedded in a model of a patient, or it could be something more complex, like for devices such as physiological closed loop control devices, which are medical device equivalents of self-driving cars, where you evaluate the physiological closed loop control device using a computational patient model. You could do testing that's completely in the computer, completely in silico, or you could do something called hardware in the loop testing, where you basically fool the real device into thinking it's acting on a real patient, whereas actually it's acting on a computational patient. So in all those cases, there is and you implement, and in all these cases, there's a real device. Second example is where the computation model is actually implemented in the device software, whether that's whether the device is software alone, or whether there's a physical device that has some software within which there's some modeling functionality. So an example of the first is the HeartFlow software as a medical device software product. That's a computational modeling framework where CT images are used as input. The software develops a patient-specific model of the coronary arteries and provides information to the clinician about the level of blockage. Another example is the CardioInsight device, which is a device that's used for non-invasive mapping of the heart, where the patient wears this vest with many electrodes that read the body surface potential, and then the device solves an inverse problem to basically reconstruct the electrical activity on the surface of the heart based on the recorded potentials after setting up a patient-specific model based on, I think, CT imaging again. So these are both kind of patient-specific models, and get into the kind of topic of digital twins. And I know that the term digital twin can be used in different ways in biological modeling. I know that there's kind of like a weak definition, as I think of it, where it's used synonymously with patient-specific models, and a more stronger definition where it's used more analogously to how digital twins are used in engineering, where there's this concept of a model that is kind of added to and provided with new data as it becomes available. So given that I don't usually use the term digital twin, and certainly this work is going to be focused on patient-specific modeling, using that weak definition of digital twin, say. Then there's this idea of in silico clinical trials. In silico clinical trials, this is a kind of emerging methodology where instead of evaluating a device on a single patient or a small number of patient models, we try and evaluate a device using a wide range of patient models, hopefully kind of representing the real indicated patient population. Might have to do a lot of sampling there. We have in OCell a project that went into depth on this idea called the Victory Project, which tried to simulate an in silico clinical trial for breast imaging devices entirely in silico, using models of the breast anatomy, compression, models of the devices, different models of pathology, and so on, and really did do a full in silico clinical trial. We also have another pathway for that use of modeling could be used in medical device submissions, which is the MDT pathway, medical device development tool pathway. This is where a company or a group comes in with a submission that's not a medical device, but a tool that could be used to evaluate a medical device. And we have a mechanism for those tools to be qualified in a certain way. And obviously those tools could be computational model tools. So that's another way that modeling could be relevant to CDRH. So I'm going to focus on this software as a medical device application. It's newer, it's much less established than this in silico device testing, but it has huge potential for medical devices, for clinical decision support systems and tools, for software as a medical device. But as we'll talk about, it hasn't really been this application hasn't really been evaluated in depth in terms of what are the credibility requirements for such applications. And I mentioned that we're going to look at cardiac models. So for those of you not familiar with cardiac electrophysiological models, this on the right-hand side is a simulation of ventricular fibrillation, just an example of simulation. Basically, they are multi-scale models. When it's done at the tissue level or the whole organ level, or at least like the ventricular level, say, they are developed from using anatomical computational meshes for patient-specific models. These are generated from MRI images. They might have some representation of scar tissue and something called border zone, which is this transitory region between healthy tissue and scar. Cardiac models also require some representation of the local fiber orientation, which impacts which direction electrical waves want to propagate in. Then they have this cellular model, which encodes how ions are transported across the cell membrane and the action potential arising at the cell level. And those require parameters that are typically not personalized. And then there are partial differential equations, PDEs, that are solved that govern the propagation. And then and they have extra parameters that are typically not personalized as well. And then you simulate that and look at some particular quantity of interest, such as the ability to induce an arrhythmia. And this is a very well-established domain. The first cardiac model was developed, I used to say over 50 years, but I've been working in the field for a while now, so I should be starting to say over 60 years. It'll be over 70 years soon, I think. Yeah, personalized cardiac electrophysiology is very well established now. Lots of potential applications. Very exciting field to be working in. And I think a good exemplar for looking at patient-study models, because it covers a bunch of different things, like developing models from imaging data, which is common for patient-study models, at least a certain class of patient-study models. It's multi-scale modeling. You can't personalize all the inputs. You have to choose which ones to personalize. And these are complex models as well. So when we started doing this work, we decided early on that we needed to distinguish between two different use cases of patient-study models. One is patient-study models used in silico clinic, used as a clinical tool. So this is where a new model is generated of a used in the field. Basically, a new model will be generated for every new patient every time it's used. So imaging data will be taken in of the patient, or generate a patient-specific model that provides some predictions, and then that's used by the clinician in some way. The second use case of patient-specific models is where you generate a virtual cohort of dozens, say, of patient-specific models from maybe hundreds if you've got a lot of data and a good framework for developing these things at scale. And then all of the virtual patients in your virtual cohort represent a real patient of some kind, or a real subject. And so you have this virtual cohort that then you can use to test a device in some way, or a drug. What's out of scope is virtual cohorts that are developed by sampling. So, for example, in silico clinical trials, where you sample parameters to generate a really large number of virtual patients. But that's out of scope for this work, because they're not patient-specific models in any way. So hopefully that this difference is clear. These are all kind of patient-specific models in the sense that they all represent a real subject. A quick overview of the timeline in terms of like where this work fits into all of the broader work that's been going on in terms of credibility assessment by FDA, by the medical devices community in the last 10 years or so. So in the early 2000s, in terms of regulatory submissions, there's little formal credibility assessment, and early 2010, there was greater awareness within CDRH, the Center for Devices, for the need for better model credibility assessment in the medical devices community in general. The ASME V&V40 subcommittee, that's American Society of Mechanical Engineers, Verification and Validation subcommittee, was formed in 2013, and they released the standard, which many or some of you will be familiar with, I'm sure. That is basically the first standard that provides a first standard for credibility assessment of computational models for medical devices, and it provides a general framework for that topic, although it is very much motivated by that kind of first use case where you have a model of a device, and generally you're reproducing a bench test, and to validate the model, you do some bench tests with the real device. It's not really motivated by patient-specific models. In OCEL, we have been doing research in this area for a while, and we formalized that into a credibility of models regulatory science program a few years ago. So we really kind of like, that really emphasizes the importance of this topic to OCEL and CDRH and FDA as a whole. I've been leading the development of FDA guidance on credibility assessment, and we released draft guidance that basically extends the VB40 standard to these other use cases such as patient-centered models, and basically tries to generalize VB40 to cover more of the different cases that we see And that was released in 2021, at the end of 2021, and hopefully the final version of the guidance will come out in 2023. But at the same time, in the last few years, we've been doing this research related to patient-specific credibility assessment of patient-specific models. So I mentioned verification and validation, and that's part of verification, validation, uncertainty quantification, which is the field that was developed by the engineering community for credibility assessment of their models, such as automotive, aerospace, structural engineering models. So, and the medical device community has been basically drawing from this field for the last few years and trying to take what we can from methods and best practices there and apply it to devices. And one of the things that I'll be looking in the upcoming slides is how does VVUQ apply to patient-specific models. So very briefly, to just make sure everyone's on the same page about these terms and the definitions, when you have a model, you develop a mathematical model of whatever system you're trying to model, and that's the mathematical model is the set of equations and parameters that are written in paper and a journal publication, and then you have a computational model, which is the software that solves those equations after implementing some kind of discretization typically. So a verification asks whether the computational model is an accurate implementation of the mathematical model and what are the numerical errors due to discretization. So it's purely a mathematical exercise. Validation asks, are you solving the right model? What is the impact of modeling assumptions and so on? So asks to compare the computational model with real world data. UQ basically takes the idea of a computational model, and instead of having single values of parameters which go into the computational model and give a single value of the output of interest, asks what is the uncertainty in those parameters, and typically represented by distributions, and you propagate that uncertainty through the model and get the uncertainty in the output, whether a distribution or maybe just a mean and standard deviation. And the UQ is familiar to even the layperson because of how weather forecasting is presented and hurricane forecast and so on. And one slide on the framework that's presented on the VB40 standard, because I'm going to talk about how that could be used for patient-specific models. So VB40 is a risk-informed framework for assessing computational models. So it asks the user to assess the risk associated with using the computational model for the particular question of interest that is being addressed. And that can be based on the consequence of making an incorrect decision, whether that's a minor harm to the patient or whether that could lead to patient death, and the influence of the model in the decision, whether it's minor because you also got animal or clinical data, or major because it's the only source of information. And then based on the risk, that determines the rigor of the credibility activities that need to be done. And VB40 defines a number of so-called credibility factors that break down the assessment. And these are different individual things to look into basically and decide to what extent you will look into that aspect of the model. So the goals of our project were to, first of all, identify unique considerations when applying, when assessing credibility of patient-specific models, unique considerations when, verifying, doing validation, doing UQ, patient-specific models, demonstrate the VVUQ process for patient-specific models, and consider how VB40 could be applied, given that it wasn't really designed for patient-specific models. Our approach was to use cardiac EP as an exemplar. We, in the paper, we first reviewed in depth the different patient-specific cardiac models that were developed, and then had a section where we considered how VB and UQ apply to patient-specific models. And we had a couple of simulation studies, which I'll talk about in a moment, and then used the review and the results of the simulation studies to finish with this, use those findings to kind of try and generalize VB40 somewhat. and specifically take the credibility factors in VB40 and ask, are they appropriate for patient-specific models? And if not, how could they be renamed, or what new ones should be added? And if they are appropriate, what is a gradation that's appropriate? I'll get to that, I guess. Some of you will be familiar with that already. So starting to go through the process, first of all, for verification, there's two parts of verification: code verification, which asks basically, have you implemented your software correctly? And calculation verification, which asks, what is the actual discretization error in your simulations? And for patient-specific models, if you recall, I mentioned there's two different use cases. Clinical tools or virtual cohorts. So either way, the methods in theory for generic models seem to apply for patient-specific models. Calculation verification, you start to get more subtlety and nuance and kind of interesting questions. And one is typically, you could do a discretization error analysis, a mesh convergence analysis on just one patient, but is that really, are the findings for one patient, which is what would typically be done, will that really generalize? And could there be risk to patients by there being some patients such that what you think is an appropriate mesh resolution from some single study, could there be a certain subpopulation of patients for which you're getting large numerical errors because you generalize some findings from a single patient? There's also the possibility that software could do patient-specific mesh resolution in the software itself every time it's run on a new patient. That's not common at all, but it's something that's possible, that kind of really ideally should be included in any kind of credibility assessment framework. Then there's a credibility factor in VB40 called use error, and that asks, like, basically what is the potential for people to have input incorrect parameters and things like that. And while that applies to patient-specific models, we realize there should also be some consideration of user variability, because there is often a stage in the generation of these models where there's some subjective decision that needs to be made. So, for example, choosing seed points for a segmentation. And then there's the question that should be asked of how variable are the results based on either inter-user variability or intra-user variability or inter-user variability. So one of the things that we looked at, to go into more detail with this question, how much does discretization error vary across patients? We investigated if we took a virtual cohort of 24 heart models that were developed by some collaborators at King's College London, and basically looked at whether it would be sufficient in general to do a mesh resolution study just using one patient, as would typically be done, or whether that could lead to misleading conclusions. And so we basically did a multi-patient mesh resolution study, generated 4 meshes for each patient at different resolutions, looked at a couple of different quantities of interest. And this figure shows some of the different quantities of interest in the different rows, or at least these two, I guess, are the same quantity of interest different ways. And this is a different quantity of interest, different meshes. And you get some idea of the variability. And we found that for the lower resolution meshes, multi-patient mesh resolution studies may not be as critical. But for the higher resolution meshes, if you did do only on a single patient, you could get misleading results, and there could be some potential for believing that you have a sufficiently high resolution mesh, where on some patients it would not be the case. So moving on to validation, one slide on this, which basically just compares with typical application of modeling and simulation for medical devices, where, like I said, for a bench, when you have a model of a device, when you do validation, to validate that model of device, you have the real device, you typically do bench testing, and generate some really good high quality experimental data that you can compare, and it's experiments that you have full control over. Obviously for patient-specific models, that's not the case, and there are various different approaches that could be used for validating patient-specific models, and these should be basically accounted for in any kind of framework. The framework shouldn't assume that a certain type of approach has been used. So a patient-specific model typically might be validated by doing a full clinical study, validating the tool output that the clinician is supposed to use. But as an intermediate stage, you might have a clinical study validating intermediate model outputs. Similarly to before, well, not really similar to before, but another possibility is that the tool does validation itself. Again, it's not really something that crops up very often. I would hope that in 20 years' time, we do have patient-specific models patient-specific model tools, software that do this, but basically they read in patient data, they generate a model, they withhold some of the data, generate a model using calibration, and then test that internally against the withheld data, and then internally lights a green light and says, I can model this patient successfully, I'm going to go ahead. I don't know if that will happen because device manufacturers are not going to want to develop tools that sometimes don't go ahead and simulate a patient, but it would be a much more it would be a higher quality tool. And then for these virtual cohorts, there are different ways that they could be validated, which validating the quantity of interest on a subsection. Maybe you have validation data for 6 out of the 24 patients, for example. So you validate on those, and then you can have confidence that you have the ability to predict for the others. Or maybe you validate a different quantity of interest if you have some other experimental data that was collected. Again, there are a wide range of possibilities for validating patient-specific models, unfortunately. It's a little bit of a busy slide, and I don't think I'll go through this in detail in the interest of time, but for UQ, in the paper, we talk about this a little bit and emphasize the difference between personalized parameters and non-personalized parameters. So there's always a spectrum in patient-specific models. Unless you have a very simple model, you can't personalize every parameter, and some parameters are not personalized and they're just fixed to population values. And then when you think, what does UQ mean? For personalized parameters, the uncertainty is just due to measurement error or calibration error. Whereas for non-personalized parameters, the uncertainty is due to population variability. So really those should be accounted for in some way when in the UQ. And the first level of UQ is to check that the measurement error is not impacting your predictions, and that should really be a critical feature of your analysis, because if you develop a patient-specific model and it's very sensitive to the measurement error of the data of the devices that you're using to collect data from the patient, then it might not give reliable predictions. And then so the second study that we did was related to uncertainty quantification for patient-specific models, and in particular virtual cohorts, and what might uncertainty quantification look like for when you have virtual cohorts and large number of patient models. So what we did was we took this paper, which is a study by our collaborators who developed this cohort of heart models. And this is a paper that basically does a simulation study where they take these models. It's a simulation study into the importance of choosing where you pace during cardiac resynchronization therapy. and the possibility of inducing a dangerous arrhythmia kind of accidentally by pacing too close to scar tissue. So they have these 24 models. Each model has scar tissue in it, and they paced close to scar tissue and far from scar tissue, and basically looked at a particular quantity, which is a proxy for ventricular tachycardia risk, VT risk, which is a dangerous arrhythmia, and basically looked at whether that proxy is, whether there's greater risk if you pace closer to scar, and that was their finding. And then we looked at, our question was, okay, is that study conclusion, what happens if you introduce uncertainty quantification? Is that, and specifically, uncertainty quantification, when you do it, it's going to change the outputs a lot. These models are always impacted by uncertainty. But is the study conclusion impacted by the uncertainty? That's what ultimately we want to show, that the study conclusion is not impacted by the uncertainty. So we tried to go through that process, and we chose one important personalized input, which is the region of, so in this image here, different colors unfortunately, but the scar tissue is this light pink, and then there's something called border zone, which is the red, which is this region where you have slightly different properties, tissue properties. It's not dead tissue, but it's damaged tissue. And that's determined from the MR images, but with a lot of uncertainty, as you can imagine, it's hard to identify scar tissue and border zone tissue from MR images. So there's uncertainty in the true border zone distribution, and so we basically estimated ranges for that uncertainty and developed new models where we contracted or expanded the border zone and repeated the study to see whether the study conclusions were still the same with the expanded and contracted border zone. And if this was the original study where basically there is a reduction in this proxy value as you pace further from scar, we did see the same. Obviously see a big difference in how the absolute values change, but we do see a reduction with the expanded border zone values and the contracted border zone, and also a reduction with the contracted border zone values. And then we also did, we considered that this figure is a bit misleading, because if you assume that the uncertainty is if it's not biased, and that some patients the border zone was underestimated and some patients the border zone was overestimated, then that's not really covered by these two figures. So we did the appropriate statistical tests, sampling from different possibilities, and in all cases the difference is statistically significant. So we showed that even though border zone distribution really affects the absolute values of the model output, it does not affect the study conclusion. So it really strengthens the study conclusion. We did that for one of the fixed parameters as well as what the border zone as a variable parameter. Okay, final 3 slides. The final section of the paper, and I'm not expecting anyone to read this table, but basically I said that we took our findings and went through VB40 and went through each of the different credibility factors in VB40 and said, is this applicable to patient statement models? And if not, how could it be changed? And that's what this table is basically. If you're interested, please go to the paper. These are all the credibility factors in VB40. These are our thoughts on whether they apply to patient statement models and the different types of patient statement modeling application. And I mentioned that VB40, if you're familiar with it, it asks users to, for each credibility factor, asks users to define a gradation of increasing rigor of activities. And it provides some examples. And so this was the example in VB40 for one particular credibility factor, the discretization credibility factor. VB40 suggests this as a potential gradation. And we, in our paper, provide what we think is a sensible patient-specific modeling version So it kind of is like a supplement. Overall, the paper is kind of like a supplement to VNV40. Okay, that's where I'll finish. Thank you for your interest. And so briefly, take-home messages are patient-centered modeling has the potential to help achieve personalized medicine, but the variability that we have to deal with leads to many challenges, both in developing these models and assessing them. And our paper is the first that we believe general publication on credibility assessment of patient-centered models. And what we've done is highlighted two different use cases, tools or cohorts. And then when we considered VB and UQ, verification is relatively straightforward to generalize the frameworks, basically. Validation is more complicated because there's so many different approaches to validating patient-centered models. And in UQ, we demonstrated how you could do UQ in certain important parameters for a virtual cohort study. And basically, future frameworks for patient-centered models will help accelerate the translation of patient-centered models from the research sphere to clinical applications. Okay, I'll stop there, and I should acknowledge my collaborators and the co-authors of this paper, Suran Galapataka, who was the postdoc who worked on this, Rick Gray at FDA, and Steve Niederer at King's College London, and Caroline Costa, who was at King's College and wrote the original paper that we worked from. Okay, thank you very much, and happy to answer any questions.
Okay, there was a question in the chat. Mitchell, do you want to ask that?
Hold on, let me get to a place where it's not as noisy. Okay, can you hear me okay?
Yes, I can.
Okay, great. So I thought it was a fantastic overview, really. I think one of the things that has come up on our end in some of our research has been related to this question of parameter identifiability, and really in the context of experimental design. So I don't know if you have any thoughts on that or any comments. You know, the two sides I've heard is that identifiability does matter if you're going to interpret your parameters, but some people would argue it doesn't matter if you're not going to try to interpret your parameters. So any thoughts or opinions on the concept of identifiability there?
Mm-hmm. Yeah, it's tricky, and so basically VNV40 and maybe the kind of literature that it was building from the UQ side, it's all focused on UQ, and there's a little bit of mention of sensitivity analysis and no mention of identifiability, but these are all interrelated topics, and identifiability is so important for physiological models with so much variability and noisy data. And yeah, I think they should be more explicitly included in these guidelines, even though arguably identifiability and sensitivity analysis are processes that you do as part of model development, and then UQ is arguably something that you do as part of the assessment. But really, and VNV40 tries to kind of separate sensitivity analysis and UQ as separate things, and that doesn't really work for patient-specific models. And that's one of the things we actually said in the paper, that they should really be linked together more. You do the sensitivity analysis and the identifiability analysis to determine which parameters are identifiable and should be personalized and impact the output. And it's very related to the UQ that you do, and it's not really kind of like separate steps. So yeah, that's something that's not really well addressed in current frameworks. Yeah, there's papers that have been written on identifiability analysis.
So I think I very much appreciated the sort of the use the use and functionality emphasis, that you wanted to do more detailed studies when you had more life-critical applications. That emphasis on variation and functionality was critical, and that was much appreciated. As I mentioned,
I think that's a very good, important part of VB40. Medical models for medical devices can be used in so many different ways that a one-size-fits-all set of guidelines doesn't really work.
Okay. Any other questions that we can answer? I think you raised a whole series of topics that are of interest to people here.
Okay, so I can see some questions in the chat. So one about, can guidelines be extrapolated to drugs and biologics as they are, or do we need major reconsiderations? VB40 and also our guidance that we released, they tried to be general for all types of applications of medical devices, or, like I've said, with the limitation, VB40's limitation that it does kind of assume bench testing. But certainly our guidance is doesn't assume validation against bench test data. And yeah, so in principle, the approaches are completely relevant to modeling of drugs and biologics. And there has been work trying to take those frameworks and apply it to pharmaceutical and biologics applications. And I know that some of my colleagues in CEDA have published in that area. And yeah, it's kind of exciting actually, because then they think about like, okay, what are the credibility factors that are needed there, or what are the specific considerations, and then present some kind of like refined approaches. And I think ultimately I hope that we will come up with some frameworks that are used across all these different medical products. And yeah, it's even being used outside of medical products as well. So there's no reason why the general framework can't be used. There might be some specific reconsiderations, but yeah, I think these guidelines can be extrapolated. And then there's a SAMD, software as a medical device, question regarding assessing credibility of models that are updated over time, such as machine learning. Yeah, that's something that we didn't consider in this, and we just stuck to a patient-specific model that's basically considered fixed, but not a digital twin, say, that is updated as new patient data becomes available, or a model that would be a model of a patient that's updated over time. You also have obviously machine learning models where they maybe have a software release with certain parameters, and the machine learning model has been tested for using those parameters. But then when it goes out in the field, they're continuously collecting data, and you can, in principle, have a updated release with improved parameters. And that's a challenge for FDA, because FDA is now having to deal with medical devices that can be updated very quickly and easily and regularly. And we don't have a regulatory framework, or our current regulatory framework doesn't really allow that. In principle, the manufacturer is supposed to come in with a new regulatory submission. There's a certain type of regulatory submission, or a subset of regulatory submissions, that allows the manufacturer to go through a slightly faster regulatory process, but still, like, that's designed for a minor design change in a physical medical device. That still might be like something that's done every few years, whereas a machine learning model could be updated every few months.
Yeah, this is Mary McGuire. I'm the one who put that forward. We're trying to deal with that now, dealing with the governance of these models and when we update them and how they are used. And right now, the current guidance is that essentially you cannot use them in anything that does not have time for the clinicians to actually sit down and go through all the details of what's involved, even though you put all the explanations in there. So apparently that effort has been essentially been blocked for the time being, because as far as I know, I don't know anyone who's doing credibility assessment on the fly. And that's, you know, for either a cohort or for an individual patient. And I was wondering if FDA was going to start to get into that, because time series models are common in other areas of engineering, but you don't run into them in, they haven't been appreciated as much, I think, in the medical area.
Mm-hmm.
So
yeah,
I appreciate if you look at that. Yeah.
Mm-hmm. FDA's digital health organization has been trying to look into trying to deal with this by coming up with a kind of a new approach to regulation where they qualify the processes that a company has, the quality control processes and things like that. And so then it's almost like qualifying the company rather than the product. And then that allows them to have some confidence that the updated products after with new parameters can be released easily. I think that's in the machine learning sphere. I'm more focused on the physics-based models, and although that this question doesn't apply as much to the physics-based models, though in the future it probably will do so. It's something that again needs to be addressed, and maybe the physics-based modeling community. By physics-based, I mean knowledge-driven mechanistic models, whatever you want to call it. Maybe we as a community will learn from whatever the machine learning community has come up with.
Okay, thank you.
You've had your hand up.
It's John Rice. I got to throw in my soapbox pitch. Like, this sounds difficult. The bar is getting higher, but industry is beginning to discover ways to start to address the issues that are going to determine whether their products are useful or not. And the academic computational community, as we explore different kinds of modeling and go into new areas, as daunting as the task may sound, if the standards are not at least reasonably understood, and people developing models do not collect the kind of information and document it as you go, then things that otherwise might be very useful models are just not going to be useful, because the evidence that's going to be needed to have confidence in those models has been lost. And it, I can't imagine what the impact of this is on the academic computational modeling community culture, if you will. That early on I was told, standards limit our imagination, and standards put limits on what we're able to do. And in the university, we're exploring, we're trying to learn. That's all true, but the reality is, for your product to be useful, it's going to have to address the kind of questions that regulators are beginning to ask, and that the profit-making medical device industry is responding to.
Yeah, thanks, John. There's an interesting question to be raised about how much credibility assessment is needed for computational models, and when do things become too hard? I obviously fully agree that it's important, otherwise I wouldn't be doing this work, and it's critical to remove the skepticism that a lot of people have about computational modeling and as a replacement for clinical data, animal data, but it can be some kind of burdensome. And actually right now what we're doing as a program, I mentioned we have this credibility computation models program, we're going through kind of collecting industry input on their challenges for computational modeling, and getting some of their feedback. And it's been interesting to hear about, when they think things are too hard and what could help with that respect. VB40 tries to help by making things risk-based so that you don't have to put as much effort in when things are used for low-risk applications, but it still provides a lot of potential work that needs to be done. And then there's arguments that people don't ask for so much for bench test data. There's someone pointed out that we do uncertainty quantification for computational models, which requires knowing about the uncertainty in the kind of experimental data that you're using to develop the model, validate the model. But often, like if you're just using experiments, there's less strict requirements about dealing with the uncertainty there. It's harder, more requirements about dealing with uncertainty for computational models. So again, it's kind of like not really symmetric in terms of what's expected of the computational model. And this is more of a cultural issue, I think, and something that will have to be addressed.
We look forward to hearing how this develops. It's great to see how seriously you're trying to work to make sure that this kind of technology is made available in medical